The, f- the first type of blood cell we'll be discussing is the erythrocyte, also called the red blood cell. So, erythrocyte is the cells of one of the cells of the blood that have no organelles or nuclei. And in humans, mature red blood cells are flexible and biconcave with only 30% more surface area. And these biconcave discs they lack nucleus and most organelles. And they have a disc diameter of about 6 to 8 micrometer or average 7.5 micrometer with thickness of 2 micrometer. Each red blood cell has hemoglobin, which is an oxygen carrying protein, and the red blood cell has about 280 million hemoglobin molecules, which each red blood cell has about 280 million hemoglobin molecules. And the adult human have roughly 20 to 30 trillion of red blood cells at any given time. So this red blood cells is an oxygen transporting cells, which I said has a diameter of 7.5 micrometer and most numerous of the form element. The female has about 4.3 to 5.2 million cells per cubic milliliter of blood, while males have about 5.2 to 5.8 million cells per cubic milliliter of blood. And as I said, the adult human in general, they have roughly 20 to 30 trillions of cells at any given time, comprising one quarter of total human body cell number. So human red blood cell take an average of 20 seconds to complete one cycle of circulation. And the red blood cells are made in red bone marrow in long bones, cranial bones, the ribs, sternum, and vertebra. And they have an average lifespan of 120 days. So this is the structure of red blood cell and hemoglobin. The red blood cell is biconcave disc with a surface with a diameter of 8 micrometer and thickness of 2 micrometer. And this is the hemoglobin, which is a bimolecules that have 4 heme group with alpha polypeptide chains, and this is responsible for transport of more than 98% of oxygen. So we are going to discuss this structure of hemoglobin also, and also the formation of the red blood cell. And these red blood cells store collectively about 2.5 gram of iron, representing about 65% of total iron contained in the body. So this is the structure of the red blood cells, the shape and the shape of the hemoglobin. So the hemoglobin, they are made up of 4 heme, each of them shown in this C, which is the iron containing heme, and they have alpha polypeptide chains. So hemoglobin, they are protein molecule. Red blood cell, I said the life cycle live only for 120 days, and they cannot synthesize new components because they have no nucleus. So ruptured red blood cells are removed from circulation and destroyed by fixed phagocytic macrophages in the spleen and liver. So the breakdown of products recycled, what we call, we are going to give us a globin amino acid, which are going to be reused. The iron will be reused, and the non-iron heme ends as yellow pigment urobilin in urine and brown pigment stacobilin in feces. So we are going to discuss on the production and destruction of red blood cell. Erythropoiesis. This is the process of formation of human red blood cells. And erythropoiesis is where by which red blood cell develop from a committed stem cell to immature cells in about 7 days. And through this process, red blood cells are continuously producing the red bone marrow of large bones at a rate of 2 million per second in healthy adults. and the production can be stimulated by hormone erythropoietin synthesized by the kidney. Just before and after leaving the bone marrow, the developing cells are known as reticulocytes, which will comprise about 1% red blood cell. So during erythropoiesis, we have the hemocytoblast that is going to be transformed into pro-erythroblast. This hemocytoblast is a stem cell that is originating from a myeloid stem cell from the pluripotent stem cell, and the pro-erythroblast will now develop into highly erythroblast. So the phases in development of red blood cell, we have one, ribosome synthesis, hemoglobin accumulation, ejection of the nucleus and formation of reticulocytes, and these reticulocytes then become mature erythrocytes. But what are the regulation of erythropoiesis? What are those that can causes formation of red blood cells to take place? One, too few red blood cells can lead to tissue aplasia, and this one can causes formation of red blood cell. And also too more red blood cell increases blood viscosity, and this one reduces erythropoiesis. And balance between red blood cell production and destruction depends on hormonal control, adequate supplies of iron, amino acid, and vitamins. And this hypoxia is an hemorrhage or increased red blood cell destruction that reduces the red blood cell numbers. And this can also be due to, and this can lead to insufficient hemoglobin, that is iron deficiency. And when there is too low red blood cell, there will be insufficient hemoglobin to carry oxygen, and that will now be reduced availability of oxygen. Example is when someone is in high altitude. And the effect of erythropoietin is that it causes more rapid maturation of committed bone marrow cells that will lead to production of more red blood cell. And this red blood cell will produce and be at the reticulocyte stage, which will be circulately in an increased manner. And they the increased circulating red blood cell reticulocyte count is between 1 to 2 days. Also, testosterone also enhances erythropoietin production, resulting in higher red blood cell count in male. So the testosterone that we have in male plays a major role in uh, increasing the number of red blood cell in male compared to female that doesn't have the hormone testosterone in quantity like we find in male. So this is a diagram to show us the erythropoiesis. Now we said the erythropoiesis starts in the bone marrow with pro-erythroblast, so that the cell near the end of the eject nodule and become the reticulocyte. So looking at this chart, some stimulus disrupt homeostasis by decreasing the oxygen delivery to kidney and other tissue. During this condition, when there is low oxygen delivery or decreased oxygen delivery to kidneys and other tissue, the receptor in the kidney cell detect the low oxygen level, and the next thing is that they increase or secrete erythropoietin into the blood. The erythropoietin secretion in the blood is signaled or is going to contact the control center, and this one will lead to the release of pro-erythroblasts in the bone marrow, which mature quickly into reticulocytes. So this reticulocyte is immature blood cell that enter into circulating blood, and they will be circulating in the blood in order to become mature to form the red blood cell that will be replacing the old or the dead red blood cell, and this will increase the red blood cell content in the blood. The large number of red blood cells in circulation will lead to more oxygen delivery to the tissues, and this one will now return the homeostasis back when, so that the kidney will now reduce the erythropoietin that will be secreted in order to reduce the red blood cell that will be produced by the bone marrow. So two major things that stimulate more production of red blood cell are insufficient oxygen delivery to the tissues, and this is due to too low red blood cell in the blood. Hemoglobin. Hemoglobin is a 4 polypeptide chain with heme in each of the chain. An ion can combine reversibly with one oxygen molecule and can also transport 23% of total carbon dioxide combines with amino acid of globin. Nitric oxide also binds to hemoglobin, and they release nitric oxide, causing vasodilation to improve blood flow and oxygen delivery. So this is the hemoglobin that has 4 chain. You have the 2 alpha chain and 2 beta chain, and it's like, and this chain is like an helix, and they have 4 ion in group that are around them. So we have 4 chain, with each chain with an in group also containing an ion. So hemoglobin is a globular hemoprotein, and they are group of specialized protein that contain heme as a tightly bound prosthetic group. The heme is a complex of protoporphyrin 9 and ferrous ion, and the ion is held in the center of the heme molecule by bonds to the 4 nitrogens of the porphyrin ring. This heme ion 2 ion can form 2 additional bonds on each side of the planar porphyrin ring, and in myoglobin and hemoglobin, one of these positions is coordinated to the side chain of histidine residue of the globin molecule, whereas other position is available to bind oxygen. So hemoglobin is found exclusively in red blood cell, and its main role is to transport oxygen from the lungs to the tissue and carbon dioxide and hydrogen proton from tissue to lungs. So hemoglobin A is the major hemoglobin in the adult, and is composed of 4 polypeptide chains, which are 2 alpha and 2 beta, held together by non-covalent interaction, and each subunit has an helical structure and a in-binding pocket as shown in the diagram previously. So the functions of hemoglobin are: 1. It helps in oxygen delivery to the tissues. 2. It helps to transport oxygen by reacting with oxygen. And the reaction of oxygen with the hemoglobin is oxygenation, not oxidation, in which one hemoglobin can bind to four oxygen molecules, and less than 0.01 seconds is required for oxygenation to take place. And beta chain move closer when oxygenated, and when oxygenated, 2 to 3 uh, dpgr is pushed out. Beta chain are pulled apart when oxygen is unloaded, permitting entry of 2 to 3 diphosphatidylglycerol, resulting in lower affinity of oxygen. So that is the two functions of hemoglobin. One, it helps to deliver oxygen to the tissue, and two, it helps to react oxygen to themselves. And that reaction of oxygen to hemoglobin is called oxidation, not reaction of oxygen to hemoglobin is called oxygenation, not oxidation. So we have one hemoglobin has the ability to bind to four oxygen molecules, and these oxygen molecules that bind to hemoglobin, when they bind, immediately they are oxygenated, they push out 2,3 diphosphatidylglycerol, and when the B chain are pulled apart when oxygen is unloaded, this 2,3 diphosphatidylglycerol is permitted to enter into the B chain again, which causes a lower affinity for oxygen to bind with hemoglobin. So 2,3-diphosphatidylglycerol, when they are present in the chain of the hemoglobin, there is lower affinity for oxygen. For oxygen to bind with the hemoglobin, this 2,3-diphosphatidylglycerol has to be pushed out of the hemoglobin. So this is a chart showing us the uh, reaction of the oxygen with the uh, hemoglobin. At this first part, the pink part, we have the oxygen binding with the hemoglobin when there is no 2,3-diphosphatidylglycerol. But immediately, the hemoglobin has been dissociated from the oxygen. We have the entry of 2,3-diphosphatidylglycerol again. So presence of 2,3-diphosphatidylglycerol will causes lower affinity for oxygen, and absence of it will causes increased affinity for oxygen. So oxyhemoglobin is the binding of oxygen with hemoglobin, while deoxyhemoglobin is the dissociation of oxygen from hemoglobin. So the process of oxygenation are: 1. Looking at this diagram, we have deoxyhemoglobin, in which the weak ionic bond and hydrogen bond occurs between alpha beta dimer pairs in the deoxygenated pairs, and this strong interaction, primarily hydrophobic between alpha and beta chains, form stable alpha dimers. When fully saturated, each gram of hemoglobin binds to 1.34 milliliter of oxygen, and the degree of saturation is related to the oxygen tension, which normally ranges from 100 mm mercury in arterial blood to about 35 mm mercury in vein. So the relation between oxygen tension and hemoglobin oxygen saturation is described by the oxygen dissociation curve of hemoglobin, and the characteristics of this curve are related in part of properties of hemoglobin itself, and in part to the environment within the erythrocyte, including pH. So this is a figure showing us the transport of oxygen and carbon dioxide by hemoglobin. 1. When the oxygen is bind to the hemoglobin, you can see the binding, and this is called oxyhemoglobin. And this oxyhemoglobin are transferred to the tissue. And when the oxygen is released from the hemoglobin to the tissue will produce carbon dioxide, which will now be bind to hemoglobin, and this is called carbamyl hemoglobin. And this carbamyl hemoglobin also help to transport the carbon dioxide again back into the lungs, in which carbon dioxide will be released from the hemoglobin to the lungs. So the other way around, the oxygen will be picked from the lungs that will bind to the hemoglobin. So that is how the red blood cell, with the help of the hemoglobin it contains, help to transport oxygen and carbon dioxide from the lung to the tissue, and also tissue back to the lung respectively. So the hemoglobin transports oxygen from the lungs to the tissue, and also transports carbon dioxide from the tissue back to the lungs. The types of adult hemoglobin. We have four major types of adult hemoglobin. We have the HbA1, which is the one with highest percentage, which is 90%, followed by HbA1c, which is about 3 to 6%. We have the HbA2, which is about 2 to 5%, and we have the HbA, which is the one with the lowest percentage, that is less than 2%. So the HbA is the major hemoglobin found in human. So this HbA is the one we found in our body. But HbA1c is the type of hemoglobin that has a glucose residue attached to beta globin chains, and the increased amount of this HbA1c is found in diabetes mellitus patients. HbA2, this type of hemoglobin first appears 12 weeks after birth and is a minor component of normal adult hemoglobin. And the last one, which is the HbF, are normally synthesized only during fetal development. This is a diagram showing us, this slide is trying to explain the formation and destruction of red blood cell. So all these stages are numbered 1 to 14. So then we are going to explain each of the stage one by one. Normally, as we discussed earlier, the life cycle circulates for about 120 days. So when their lifespan is reached, at stage 1, the red blood cell will die and undergo phagocytosis. This phagocytosis is done by the macrophages in the spleen, liver, or red bone marrow. So in this red blood cell, when they are phagocytosed, the hemoglobin, which is the major component in this red blood cell, will be broken down into two parts. We have the globin and the heme. The globin is the protein which is going to be broken down to form the amino acid, and this amino acid will be reused for other protein synthesis. So without the globin, part of the hemoglobin has been used up. So the globin will form the amino acid which the amino acid will be used for protein synthesis. The second part of the globin is the heme, and this heme will also undergo two processes. Firstly, from the heme, the heme will be broken down into iron, 3 iron, and bifadine. So in the process known as erythrocytes. So the iron will be acted upon by transferring. This transferring will transfer the iron into the liver, and they will use it to form ferritin. And this iron also will come out from the liver. This will form of iron 3 iron again, and will be transported through transferring again into the red bone marrow for another process of blood cell formation, which means the iron that is removed from the heme are still going to be reused back in the bone marrow for another production of red blood cell. And the bifadine that is left in the heme part of the hemoglobin will now be converted or used into plasma, and will circulate into liver band to albumin as bilirubin. So this bilirubin will now be transferred to liver. From the liver, they enter into the small intestine, because this bilirubin is a pigment that need to be excreted from the body. So in the small intestine, the bilirubin is acted on by the bacteria and form what we call urobilinogen. So what are the ways through this urobilinogen will be removed from the body? One, some of this urobilinogen will enter to the kidney and will now become urobilin, which will now be released as part of urine. While some of the urobilinogen will enter into the large intestine and they will now become stacobilin that will now be added to our feces. So in that number 8, which I said the iron will be transported from the liver into the uh, bone marrow, that is found in the bone. So the iron 3 iron will combine with the globin, which is the amino acid that has been gotten from the hemoglobin part, with vitamin B12 and hormone erythropoietin will now regulate the production of another red blood cell, which will now be released again to circulate for about 120 days, and when they hit their lifespan they start the process again. So the formation is done in the red bone marrow with the help of the iron 3 plus, globin, which is the part of the hemoglobin that has been gotten from the former destruction of red blood cell that was taking place, and we have the vitamin B12 and erythropoietin that regulate the production of red blood cell. So please take your time to understand this chart that is explaining to us how red blood cell is formed and destroyed in the body. Red blood cell lifespan and circulation. The red blood cell are replaced at a rate of approximately 3 million new blood cells entering the circulation per second. So damaged or dead red blood cells are recycled by phagocytosis, as I've discussed earlier in the previous slide. These red blood cells, when they reach their lifespan of 120 days, they become damaged or dead and they are going to be recycled by phagocytosis. And phagocytosis is done by the macrophages in the liver, spleen, or bone marrow. So the components of the red blood cell, which is the hemoglobin, are the ones that are individually recycled. And this hemoglobin are broken down into two. You have the heme and the globin. The heme part are stripped of iron. Iron are removed from the heme, and the remaining part of the heme are converted to bifidin, which is then reduced to form the bilirubin. So this iron that is stripped from the heme is recycled by being stored in phagocytes or transported through the bloodstream bound to transferrin. So this diagram, we have the macrophage in the spleen of liver and bone marrow. They remove the iron from the in part of the hemoglobin, and this iron are transported in circulation by transferring into bone marrow to initiate another red blood cell formation. Also, the remaining part of the heme are turned to form the bifidin, while some amino acid are also gotten from this part of the hemoglobin. This amino acid are also transported back to red blood cell formation to also initiate red blood cell formation. So the bifidin that's gotten from the heme are reduced to bilirubin, which are then transported to the liver and they are excreted in bile as bilirubin. So this bilirubin derived products are called the urobilinogen, are going to be transported in form of urobilin as tecobilin, which are going to be eliminated in feces and also in urine. So the red blood cell formation is done in the bone marrow with the help of iron 2 iron and globin, and this one are the one that will now produce red blood cell, which will now enter into the circulation, and they are going to continue their lifespan for 120 days. So this has shown that we have come to the end of the first section of the red blood cell. So try and use this question to guide yourself and see whether you are able to understand some of the things I've discussed about the red blood cell. And when you are through, you can click to check your answers. So this is the answer for the question on the previous slide.